Hello everybody. I'm just going to quickly run through some of the lecture content and just give a bit of an overview of some of the key concepts and some of the things that we were talking about. This is not meant to be a crash course in molecular biology. A lot of what I say you will need to reference the original lectures and videos to really get the context and the understanding, but I'm just kind of summarizing a few key points here. Okay, but a lot of these summaries don't make a lot of sense unless you know the information that I am summarizing. Okay, so it's important that you go back to the original lecture content, and your primary source of study is the original lecture content. But having said that, I'll just quickly whip over some of these slides and just talk about some of the concepts. So the first lecture was on predominantly colon cancer. And towards the end of it, we talked a little bit about metastasis and angiogenesis. So in this series of lectures, we're using colon cancer as a primary example. And we talk about, there's a lot of things we can understand from colon cancer that apply to all cancers. So it's a good model and it's been well studied. So there's a little slide example here showing an image of these villi and on the lining of the colon. And just to give a bit more clarity to that, I included this slide here, which shows a profile of one of these little these crypts, and it highlights these stem cells at the base, and these so it highlights at the base here are the stem cells. And these stem cells are constantly growing and dividing, and when you get mutations in these cells here, they give rise to those mutations being propagated in these transient amplified cells. And as more mutations occur in the stem cells, then over time these short-lived transiently amplified cells, as they're constantly being replenished, as they die off and are replenished and die off and replenished, being sourced from the stem cells, then the mutations that occur in the stem cells end up being displayed in these transient amplified cells. So and that's a concept we talk about later when we look at clonal selection. So I thought it was good to include a slide that's not from the textbook that just talks to this slide a little bit. Okay, so we're talking about the accumulation of mutations within a cell, and about if it's a rapidly dividing cell, it's giving rise to other cells that have that same mutation. So we're talking about how cancer evolves, whether it's a single mutation or multiple mutations. And I think it's pretty obvious that it's multiple mutations, but when people started to think about this the first time, it wasn't obvious. So we talk about multi-step tumorigenesis, and we're just talking about this accumulation of mutations within a genome, within a cell. And we look a little bit about the time frame of this. I mean, is cancer something, if you suddenly are diagnosed with a cancer, is it something that occurred and developed over a short period of time, or is it something that's been developing over a long period of time? So we looked at some statistics and some epidemiology of cancers, and we looked at the incidence of lung cancer, and we were just noticing in these things here that you've got the occurrence of lung cancer over a period of time, and you can match that to the increase or uptake in smoking. And if this was 1930s in the 1970s, then the uptake of smoking is mirrored by the development of cancer. So this is clearly indicating a long time frame from the exposure to the occurrence of the disease, which gives a bit of a hint about what's going on. Also, I guess when you're thinking about this process, you can think of we know the frequency of mutations is quite high, so it's the accumulation of mutations over a long time period. I guess one way to think about this is we've got a large genome and mutations occur sporadically throughout the genome, and you need to have that, coincident of a mutation occurring in a gene that's involved in cell regulation, cell cycle growth or cell cycle proliferation, differentiation, one of those, apoptosis, one of those important processes. A key gene, eventually, as mutations occur, will pick up a mutation. Okay, and then we talk about whether those mutations are oncogenic mutations, in terms of hyperactivating an oncogene, or whether those mutations are mutations that inactivate a protein. And then we talk about tumor suppressors. So we talk about loss of function of tumor suppressors and gain in function of oncogenes. And don't forget, we also talk about there were 2 alleles for each gene in the genome. With oncoproteins, hyperactivation of a single allele is rare, but is enough to give a change in phenotype with more growth. Whereas with tumor suppressors, there were, again, the 2 copies, 2 alleles. You need to knock out both copies because the remaining good copy can protect the cell from the loss of function. Okay. So again, you need to, when you're looking at oncogenes and tumor suppressors, you need to think about the genetics of them, 1 allele versus 2, and what's the consequence of having mutations in those scenarios. Anyway, we'll talk about that later when we get to that stage. We also talked about looking at various cancers which all follow this idea of over a long period of time increasing incidence. Again, this is going back to the idea of picking up multiple mutations over a period of time. Okay, we then went to look at the age of death from common types of cancers, and I think the take-home message there is that it's not related to it's not primarily related to your socio-economic group or where you live or those kind of things, but it's to do with the accumulation of mutations, and it's genetic, and there's a lot of similarity in the occurrence in different parts of the world. We also looked at the rate of progression, and if you're exposed to a mutagen, then the rate that is converted into maybe a cancer or tumor progression, it's the same regardless of whether you're exposed at a young age or an old age. It's the rate of progression is based on the accumulation of mutations driven by the mutagen. Okay. The textbook then starts to look at various biopsies taken from patients with colon cancer, and this is one of the advantages of studying colon cancer. You can do, colonoscopies and look at biopsies, and they started to look at the development of the cancer, and they started to notice the occurrence of polyps, and it turns out polyps were the early stage of cancer development. Okay, and they started to formulate this idea of a succession of phenotypes, thinking that maybe that over a period of time that we have this situation here where we have a tissue that's picked up a cell that's picked up a mutation, and because these are rapidly growing cells, therefore this cell is growing at a faster rate than the normal cells. So by the time this cell, let's make some general assumptions here, by the time this cell doubles to give 2 cells, this cell might have doubled to give, say, 3 cells, for argument's sake. So the rate of growth of this has been slightly enhanced by the mutation it's accumulated, and over a period of time these cells will outcompete the normal cells. So these cells will start to dominate the population, and that's what's being shown here. Okay, and then And then within one of these cells, which is originated from these cells, another mutation occurs. And that gives it a growth advantage over the surrounding cells. So this one then starts to dominate. How can we sort of represent this? Let's think about this. So let's assume we've got a biopsy of a tissue here, and they've detected an unusual cell here, okay, with a mutation in it, as opposed to the normal cells, okay. And over a period of time, that cell has a slightly enhanced growth phenotype, so it simply grows and divides at a faster rate than those around it, okay. And then within one of those cell types, it accumulates a different mutation, a different phenotype, okay. So this will now start to outcompete against the parent cell and the normal cells. So this one will start to grow and outcompete those around it, okay, and so forth. And this process occurs up until the point where the cell line has picked up enough mutations within it to give it a growth characteristic that, or other characteristics are affected in cell biology, so that it can begin to metastasize, and it can begin to release factors to draw in blood vessels to it, so that it can keep growing, and we've got nutrients and oxygen exchange, and at some point it's able to survive in isolation from those around it. And at that point, it's able to leave and metastasize to another part of the body. Okay, so again in the lectures we talk about some of the factors that control angiogenesis, some of the factors that are involved in metastasis, and some of the factors that are involved in the destination tissue that make it a good home for the new cell. All right. So again, there's a whole bunch of stuff in the lectures concerning that process. So this generally, this change in phenotype over time is referred to as a succession of phenotypes. Again, we're often looking at the same thing from different perspectives. So we're looking at mutations within a cell at a cellular level, and changes in mutations of DNA at a cellular level. But as that cell is growing and dividing and can be observed, then we see a change in phenotype. So then we talk about a succession of phenotypes over time, okay? And then as these phenotypes are things that we can recognize in terms of polyps and adenomas and all these late stage cancers, we can then talk about the evidence that shows that we're getting these multiple mutations within these cells. Okay, so and also we talk about clonal selection, which is again cells that have picked up mutations that have growth advantages compared to those around it. So in a lot of ways it's similar concepts, but looking at different scales. All right, and if you understand a process at multiple scales, then you have a much better understanding of a process. Okay, so the other things we're starting to look at here. So this succession of phenotypes, it could have been other things that were going on at the time. So they had to prove that it was a succession of phenotypes. So using colon cancer, they looked for evidence that what they thought was the most logical process was actually happening. All right. So some of the evidence they've got here, I've got 3 points of evidence, first, second, third. So one point of evidence was that when you're looking at normal cells, so you've got these normal cells here, that at some point there's been a mutation of some description, and there's a growth of cells here which are growing at a faster rate than those around it. All right, so this might be some small growth that's observed, I don't know, a polyp of some description or something. And then from within these cells, additional mutations have occurred, and then there's another growth here which seems to be emerging from the first mutation to give the second sort of observable phenotype, these cells that have got a different characteristic. And then likewise this can give rise to another pattern of growth here. So it's a case of from the original normal cells, there's been a mutation leading to growth here, more mutations leading to the more severe phenotype, and more mutations leading to what may become metastatic or a problem for the body. Okay, so these were observed in biopsies taken from people with colon cancer. Okay, but to go from normal cell to the first stage might have taken a year or two to observe that growth, and then there might have been another stage that takes a couple of years to observe, and then another stage that takes years to observe. And obviously when you take a biopsy from someone, it's a snapshot. It's not something you observe over a 3-year period. So they were observing this phenomena, which makes sense in terms of the succession of phenotypes, but they're not actually studying and observing one batch of cells that are growing into malignant phenotypes over time. So you can't just say, well, I've observed that, therefore it's a succession of phenotypes. All right? It's a piece of evidence that lends weight to the hypothesis. All right? So don't think of things so simply, but it's a good piece of evidence. All right? There's a second piece of evidence here where they were looking at people. So they were taking colons from different patients, and if a patient has these little polyps that are starting to develop, then over time these were leading to full-blown cancers. Okay, the dreaded C word. All right. Whereas people with less polyps were getting less occurrence of cancers. Okay, so that is saying that over time these things must be developing into more full-blown cancers. Okay, just because they're present and over a period of time. So again, look at the slides for a more detailed description, but that's the kind of other evidence we were looking at. All right, and then the third piece of evidence was that there's a disease called familial adenomatous polyposis, so FAP. Okay, and as part of that disease, there's a gene called APC. All right, now this gene turned out to be a tumor suppressor, and it's commonly found in people with this disease, and therefore you can start to relate the occurrence of a single hit, a mutation in the disease, which is important for understanding multi-hit hypotheses and all of those processes. But also you can study those patients, and they have many more polyps in the lining of their intestines than normal people because of this mutation. All right, and then from these studies there's a higher incidence of cancer. All right, and again that's lending weight to the idea that people are more prone, and therefore it's a succession of phenotypes over a long period of time. Okay, so there's a lot more evidence out there, but the textbook highlights 3 pieces of evidence. So have a look at those bits of evidence and have an understanding of why they're important in looking at this, of solidifying this concept of multi-hit mutations, but in particular succession of phenotypes. Okay, so we're looking at it at a phenotypic level here. Okay, and then they studied this in colon cancer, and they also then started to look at other types of cancer, and it's a common theme. It's not something that's unique to colon cancer, but doesn't help us with the understanding of other cancers. All right, so I guess then the important thing is to say, well, we know it's the accumulation of mutations. We know one of those mutations is APC. We've got to understand what those other things are. Okay, what are those other things that are mutating? All right, and this led to the development of ideas about gene function. So we have this class of proteins that were initially discovered called oncoproteins. Okay, and RAS was the big one, SRC was a big one, receptor tyrosine kinases, they're big ones. So there are classes of proteins that have individual functions in the cell, but we can generally call them oncoproteins. And the characteristic of these things is that they can pick up mutations in one copy of the genome. So one allele of these oncoproteins can pick up what's called a hypermutation, and it makes it locks that particular protein in its on state. And that's a pretty vague statement, but if you look at an individual protein, you can understand what that mutation is that locks it on. Okay, and so if you look at RAS, you can look at which point mutations, knock out the ability of RAS to hydrolyze GTP. If you look at the receptor tyrosine kinase, you look at mutations that lock the receptor in a structural form that is constantly signaling. Okay, so for each protein, there are different ways in which these point mutations can lock things in the on state. And if you can do that to a protein, then it's an oncoprotein, and it's driving growth. Okay, so people started to think about oncoproteins a lot when they studied cancer, and then later on they discovered tumor suppressors, and that came out of the study of retinoblastoma, the disease. Okay, and they discovered a protein which they called retinoblastoma, and a gene which is the retinoblastoma gene. But an understanding of how that protein works is pivotal in understanding how many cancers work, even though it was identified in a disease of the eye, which is retinoblastoma. But it's also identified a new class of tumor suppressors. Okay, so then they had not only oncoproteins but tumor suppressors that have a role in cancer. Okay, and then we can talk about that as we get there. All right, so let's keep going. So when you look at the breakdown of types of mutations that are observed in these cells, they notice that more tumor suppressors were inactivated than the number of oncogenes that were hyperactivated. And that's because it's harder to hyperactivate a protein. You're relying on a single point mutation to enhance its function. And that's a very rare event, but it does happen over time. Whereas with tumor suppressors, any of your kind of bog standard insertion, deletion mutations are going to throw the reading frame of that exon, and it's going to that protein is going to lose its function because it's going to be inactive. Okay, and once you start to lose a tumor suppressor function, that's going to allow the cell to grow. And because those kinds of mutations are more common, then these tumor suppressors tend to outnumber the number of oncoproteins. We then start to look at so we've talked about this succession of phenotypes in a fairly generic way, looking at those cartoon diagrams. This is just showing a succession of phenotypes within a patient who has colon cancer. And you can see here that you go from a small polyp to a larger adenoma to a mass of these tumors that can become malignant. And we know that during the stages of that transition that we're accumulating more mutations, leading to more severe phenotypes. So again, have a look at those lectures slides to have a look at that. We talked about this idea of common or distinct genetic mutations within cells, and what we tend to see is that there's a handful of common of likely suspects, if you like. So it's quite likely if you have colon cancer that you'll have a mutation in the APC gene, because we know that from familiar studies that it's something that's quite commonly observed. You may also have mutations in a tyrosine kinase, some other protein that we don't know about, and a p53. And that might be some of the key mutations that were observed in those cells. Otherwise, you might have a mutation in the APC gene and the RAS or the RAF protein and some other uncharacterized protein leading to tumor. So not everyone has the same mutations in a colon cancer, but there's a bunch of likely candidates which tend to be seen frequently, and each individual biopsy has a mixture of these different mutations. Okay, so there's 35,000 genes in the genome. Maybe there's a couple of hundred that are particularly important for controlling cell growth, and out of those selection of a couple of hundred, there might be 10 or 20 that are more common within a cancer type, and and a particular patient might have that subset of 10 as opposed to that subset of 10. Right. So there's all these sort of likely candidates that occur again and again. And we look at some of the processes and functions of these likely candidates. So that's why we then go ahead and look at receptor tyrosine kinases and RAS and a bunch of signaling pathways, because these likely candidates are interact together within the cell. Okay. There's this concept of another way of thinking about succession of phenotypes or multi-step tumorigenesis, multiple hits within one cell. Another way of thinking about that is looking at clonal selection. So this is the idea that you've got a whole bunch of cells, and one of those cells picks up a mutation, and that particular cell over a period of time is able to outcompete and outgrow the ones around it. So you start to have a higher proportion of these, okay? And then within one of these cells, another mutation occurs, so that over time, this one now can grow at a faster rate than those around it, all right? So you tend to get a new population which is outcompeting the initial population. So the idea is, if you think about Darwinianism and evolution, it was this idea of survival of the fittest, that some species picks up a mutation that gives it a phenotypical characteristic that allows it to survive in a situation in which the others can't. What's a really silly example? Maybe a mutation that gives a giraffe a slightly longer neck. Some sort of point mutation that leads to a growth characteristic or a developmental change. So you've got animals with a slightly different characteristic. If at some point there's a drought and only those top level of trees, those leaves at the very top, the animals that can eat those ones are going to survive, and the shorter ones are going to die off, and therefore you get a shift in the population of the genetic pool, giving rise to animals with these different characteristics. So droughts wipe out populations of organisms, and those that have got a ability to survive the drought through some sort of mutation, they then tend to become the dominant population. So there's all these. I don't want to get into Darwinian evolution. I'm not an expert on it, but that idea of survival of the fittest, that those models of thinking have been applied to thinking of survival of cells and having characteristics that allow you to outcompete the surrounding cells. So it's kind of nice to take this, because evolution is driven by mutations, so it makes sense to try and think about mutations in that context. This idea of linear clonal selection is a fairly simplified, model. It's not the best model. You can then start to consider multiple selections of mutations existing simultaneously, giving rise to slight variants of cells, so that there is some heterogeneity within that population. So initially, when we're thinking about clonal selection, we're talking about a group of cells picking up mutations, and it was like the same cell type. Whereas in actuality, there's a range of different mutations accumulating within that population. But typically, the mutations that are occurring hark back to this idea of coming from a common subset of genes. Okay, and then I guess there's then a bit of a breakdown of some of the common players that are common to colon cancer. Okay, so biopsies from one patient may have RAS and P53 and PRB, whereas biopsies from another patient may have ABC and P16 and one of the SMAD genes. Okay, but these are the common players that are involved in driving growth or slowing growth, and mutations in those are the ones that are important for developing cancer. We then went on to talk about metastasis and how other phenotypes, not just growth, but other sort of taking over other cellular processes are important in cancer as well. Obviously, growth is the easiest one to visualize without an understanding of a lot of cell biology. But as you know more about cell biology, you can start to appreciate how mutations in pathways relating to apoptosis are important, or mutations in pathways relating to growth factors that might encourage blood vessels to grow towards the cells. So there's various factors that can be released by these cells, which manipulate the environment to its advantage. Okay, so then we talked about how in some cells you may have mutations in cadherins, or you might have the release of peptidases that can cleave these cadherins, or that can cleave the extracellular matrix. So all these properties of cells are slowly changing to allow these cells to exist in isolation. Okay, so they're no longer, the growth is no longer inhibited by the neighboring cells, and they can exist in isolation. So again, I don't go into this in any great detail, but if you go to those lectures, you'll see there's a discussion of some of those properties. And then we also kind of link this through to metastasis. So angiogenesis and metastasis are kind of related, and once the cell has picked up these load of mutations, it can then start to manipulate growth through the extracellular matrix, escape into a blood vessel or a lymph system, and then leave the system and invade neighboring tissues. Okay, and some tissues are more susceptible to invading cancer cells than others, and again we talk about some of those concepts. So just have a look at some of those slides and some of those concepts. All right, so angiogenesis and metastasis are kind of related. All right. Okay, so we then went on to lecture 2, where we talked predominantly about cellular oncogenes.